Welcome, everyone, and thank you for joining us for OA Week 2023. My name is Kalina Grab, and I am one of the GOA-ON secretariats. I am at NOAA Ocean Acidification Program as a Knauss Fellow in International Policy. And please note that this session is being recorded. OA Week is presented by GOA-ON, the Global Ocean Acidification Observing Network. And GOA-ON has three different sponsoring organizations. First, NOAA, the United States National Oceanic Atmospheric Administration. Second, IAEA-OA-ICC, the International Atomic Energy Agency Ocean Acidification International Coordination Center. And third, the IOC UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. GOA-ON was established in 2012 with just a handful of members, and since, GOA-ON has grown immensely. It now has over 950 members from 114 countries, and it also consists of 9 regional hubs, which span across continents and oceanographic regions. We'll be hearing from most of these different hubs and regions throughout OA Week. If you're not a member yet, you can join GOA-ON today by visiting goon.org. OA Week debuted in 2020 and returned in 2021 when events and conferences were postponed due to COVID-19. Following the successful in-person symposium on the ocean in a high CO2 world in 2022, GOA-ON is bringing back OA Week 2023 to maintain momentum around OA research and provide a virtual platform for the ocean acidification community to exchange their latest findings. We are thrilled to present a range of the ocean acidification topics and speakers from around the world. During the presentation today, all participants are in listen-only mode. You are welcome to type any questions into the question box, which can be found at the bottom of the control panel, which may be on the right side of your screen. We'll be monitoring incoming questions. Directly after each presentation, we'll ask 1 to 2 questions per speaker, and then we'll pose the rest of the questions to all of the speakers during the question and answer section, which will begin immediately after the final presentation. For discussion, you can also use the raise hand function in the toolbox at the bottom of your screen, and we will call on you to ask your question directly. And with this, I would like to introduce your moderator for this session, Austin Pugh. Austin's interest in ocean acidification is rooted in his background in paleontology and the change of the planet through time. Austin is the Climate and Ecosystem Coordinator at the Northeast Regional Association of Ocean Observing Systems, where he coordinates the Northeast Coastal Acidification Network and is the Community Manager for the Ocean Acidification Information Exchange. Austin is also the Northern Hemisphere lead for GOA-ON's ICONEC community, and is excited to see the community of early career ocean professionals and GOA-ON becoming more active in the organization. Austin, the floor is yours.
Thank you, Kalina. Welcome everyone to the ICONEC session of OA Week. The way that we're going to work things today is we're going to do all the presentations. I will be introducing some of the speakers. Then they're going to be on the hot seat for a couple of questions. For at the end of today, we've put aside some time to do a more general discussion where you can either ask the speakers more questions, or we can have a bit more of a general conversation around ICONEC, who we are, and where we're going. But to lay the groundwork for that, I have a very brief presentation about who we are as the International Carbon Ocean Network for Early Careers. That's what ICONEC starts for. Next slide, please. So just a bit of background where we came from. The idea of ICONEC came from the 5th International Symposium on the Oceans in a high CO2 world, which happened in Lima in Peru. And at this meeting, there was a side meeting for GOA-ON hubs. And one of the main topics at that meeting was the lack of early career people that were in the room at this GOA-ON meeting and that were part of the makeup of GOA-ON. I was lucky enough to be part of that side meeting. So I was in the conversation from the beginning. And then over the rest of that conference, the ECOP members there really made this organic effort to band together and create a network amongst themselves, which resulted afterwards in our very first virtual meeting on October 24th. And you can see the pictures, a picture of all the attendees there. There's just about 20 off to the right. After this, we've held about 6 community meetings, and we've had a steering committee that's met 12 times. So we've really had some momentum as far as membership goes. Next slide, please. And now, today, we have 178 members, which represent 35 different countries. If you could click, Kalina. Those are all the countries that are represented. However, this is not an exhaustive list of the countries we want to have involved. So we ask you to share ICONEC and spread the word. A network is only as strong as its own members. So we're trying to expand and make sure that all GOA-ON ECOPs are involved with ICONEC, as it's this invaluable network. Next slide, please. And the driving force behind it is the steering committee. You can see myself and Margaret Ogandare as the Northern and Southern Hemisphere co-leads respectively. If you could click, Kalina. And once more, we have Sarah Cryer, who's presenting today, and Natalie Bravo as our social execs. Click twice more, please, Galena. Natalie's doing double duty with Mary Margaret Stoll as our secretaries, and one more time. And finally, we have Anna Hughes, who's our brand new, this just happened about last week, North American Hub Liaison. So this is a new thing we're trying as ICONEC, and that's to have a member of our steering committee sit on the steering committee of every other hub. So we're trying it out with the North American Hub, as they were the hub that approached us. But if you are interested in getting involved in not only the ICONEC steering committee and leadership, but also potentially become more involved and create closer ties with your own home hub, please let us know. We are currently looking for members for every other hub that is in the North American hub. Next slide, please. And the community goals around ICONEC, this is what drives our community. So we've done a little bit of the framework, and now I want to give you really the meat of who we are and what we do. And that's to provide ECOPs opportunities to increase involvement with local GOA-ON hubs. As I mentioned, this is GOA-ON's goal, is to create ECOPs that come through the ICONEC network. And then by the time they leave the early career stage, they're already integrated and part of their home hubs and are actively contributing to the network that is GOA-ON. Also to facilitate the knowledge sharing capability between ECOPs, so create a network of peers. One thing that COVID-19 did, which was really unfortunate, was prevent that face-to-face meeting in our early career stage, which definitely stunted some of these peer-to-peer relationships that we could have had. And ICONEC hopefully can synthesize that and move these relationships on a little bit faster, as a lot of the ECOPs today are a little bit behind where we would like to be with these professional relationships. And finally, to create and promote opportunities for ECOPs to engage in relevant projects. For example, we are currently in the middle of creating a perspectives paper on early career perspectives of OA. That's currently being written. You won't be able to hear more about that from any of our presenters today, but we could speak a little bit more about that in our question and answer session. Next slide, please. And finally, this is just a little infographic that shows how ICONEC works with the GOA-ON hubs. And that's we're this connective tissue that lines between all of them and creates communication back and forth. So ICONEC members are also GOA-ON members. They're GOA-ON hub members. It goes back and forth, and it creates a bit more unity between the hubs at the ECOP level. Next slide, please. I just want to say thank you for listening. Please share ICONEC. I'm not going to take questions now because we have lots of time at the end for discussing ICONEC, and this was just supposed to give a little bit of background to this session. Instead, I am going to move on, and I'm going to introduce our very first speaker, Sarah Cryer of the National Oceanography Centre and University of Southampton in the UK. Sarah's recently completed her PhD titled River to Reef in Organic Carbon Cycling in the coastal zone. in which she examined the influence of river water on the interplay between the ecology and biogeochemistry of tropical coastal environment. She employed a multidisciplinary approach, including fixed point observatories and autonomous surface vehicle equipped with biogeochemical sensors and discrete water sampling to investigate drivers in coastal pH and CO2 fluxes. She's currently in the process of moving back to Scotland, where she's hoping to find a postdoc with ocean carbon or biogeochemistry research. Please take it away, Sarah.
Thank you, Austin. I hope you can all see my screen. It's great to see so many people joining our session here today. So I, as Austin's nicely introduced me, I'm going to be talking about one of the chapters of my PhD, where I was looking at what's controlling PCO2 and supersaturation of PCO2 in a shallow river-fed tropical coastal ocean environment. Oh, I think it might skip a slide, it's a little bit slow. So in the open ocean, air to sea CO2 flux is the major control on ocean pH and PCO2, along with seawater temperature. But in the coastal ocean, there are additional processes which will influence pH and CO2 flux. And this includes ecosystem metabolism, so the balance of photosynthesis and respiration. So if there's lots of respiration occurring, we're going to have a lot of CO2 production, driving down pH, and photosynthesis the reverse. Water residence time will play a role that can influence how much, or it will impact the influence of the ecosystem metabolism, and also riverine inputs of CO2 and organic matter. The organic matter can then be respired, producing CO2, and finally wind speed can exert a critical control on CO2 fluxes. So my main question is for this chapter was what is controlling pH in a tropical environment where we've got freshwater input, we've got seagrass beds, and we've got mangrove forests. So to do this, my study area was located in Belize. So Belize is located in Central America, just south of Mexico, and we're focusing on the area around Belize City. So north of Belize City, the Belize River discharges, and this drains the largest catchment within the country. And so we have the Belize City Peninsula. We then have the river mouth to the north. We have extensive seagrass beds that go out towards these mangrove-covered islands. And then we have the Belize Barrier Reef, which is the second largest barrier reef system in the world. And this is just a schematic of my area. So we've got the river that's going to be bringing in CO2, organic matter, dissolved in organic carbon, inorganic carbon as well. We've got seagrass beds, and then we've got the mangrove keys, which might also be a flux of organic carbon and inorganic carbon into the coastal ocean. And I'm interested in what is the CO2 here, and what is the pH, and then what is this flux of CO2 between the atmosphere and the seawater. So initially we looked at salinity. So this is data collected using a sensor mounted on an autonomous surface vehicle. And we've got our whole study area on the left here, and then two transects on ebb and flood tide. And we've got low salinity. You can see where the Belize River mouth comes out, north of Belize City. We've got low salinity and a little bit of low salinity to the south of the peninsula as well. But quite quickly, the low salinity water is mixed away. And by the time we reach past the Belize City Peninsula, we're into high salinities. And this kind of is the same on ebb and flood tide, with flood tide low salinity water discharging a little bit further, but not reaching out to those mangrove covered islands or the barrier reef. So what we want to know is what is driving the CO2 outgassing. So if we look at the flux of CO2, everything that is a red mark point means that there is a positive flux from the seawater to the atmosphere of CO2. So this is showing that across the majority of our study sites, we have CO2 outgassing. And this isn't linked to the salinity of the coastal ocean. So it's thinking this isn't due to freshwater input, but due to other processes that are occurring. So first of all, we can look at dissolved oxygen. So here I've shown apparent oxygen utilization, which is the difference between measured oxygen and the saturated dissolved oxygen saturation based on temperature and salinity. And where we have red points, that means that the oxygen is supersaturated. We can see across the majority of our study site that there is supersaturation of oxygen. Close to the river mouth is undersaturated, which kind of be expected as there's a lot of organic matter coming in, a lot of respiration, a lot of microbial respiration likely taking place, which is driving down respiration is driving down oxygen. But across the majority of the site, we're supersaturated. So this suggests that there is large amounts of photosynthesis occurring. Sorry, my screen is not moving in the right direction when I click the buttons. Okay, here we go, sorry. So this suggests that there's very high rates of photosynthesis occurring. So then we can look at the inorganic carbon isotopes. They can provide insights to other processes occurring. So in this region, I was using inorganic carbon isotopes, so delta-13C DIC, and organic carbon isotopes, so delta-13C particulate organic carbon. And this is just a simple schematic just to kind of give you a brief introduction. But to begin with, inorganic carbon isotopes are going to be relative to it's a relative scale. So if we start with the inorganic carbon, if it becomes more positive, this means that there is degassing and photosynthesis occurring. And if it's more negative, we're going to have respiration, ingassing, and weathering. And organic carbon is a little bit the opposite, because the respiration is going to make it more positive and photosynthesis more negative. So the delta-13C signature was depleted relative to conservative mixing. So we can see, and we have this spatial map close to the river mouth, it's more negative, which is what we would expect. And as we get closer to offshore, it becomes more positive. And this is what it would be expected. A river water is going to have a more negative signature than the open ocean. However, when we look at our conservative mixing on the right-hand side plot, we can see that the conservative mixing is shown by the gray shading. And we have our data points below, and the majority of our data points are more negative than we'd be expecting from conservative mixing. So this suggests that respiration is dominating, and the respiration signal is greater than the signal of outgassing and photosynthesis, because it is making our delta-13C more negative than we would expect. And this correlates well with our high PCO2, where we have a lot of CO2 in the water column. So that suggests that respiration is driving this. We can also look at what the inorganic and organic carbon isotopes can tell us together. So to begin with, I've plotted just organic carbon against inorganic carbon isotopes, and we can see that there is clusters of different groups. So I've colored these different groups so that we're just able to identify them on the map. And you can see that, like, the ones that have a more ocean inorganic carbon signature of around 0 are further out, and the ones that are more negative are further in. But there is this different relationships in different areas. And so organic carbon isotopes can be very powerful when used in combination with the organic carbon to nitrogen ratio, because different organic matter will have different signatures. So when we look at our, if we now put those same groups onto this plot where we have carbon to nitrogen ratio against organic carbon isotopic signature, you can see that they fall within different organic matter sources. So all the green ones fall within the seagrass. We have some falling within mangroves, and the majority being in macroalgae and phytoplankton, and these are the ones close to the river mouth. So this suggests that our three main sources of organic matter in our region are mangroves, seagrasses, and macroalgae and phytoplankton, and in fact are not the terrestrial plants that we might expect to be coming in from a river. And when we look at this spatially, we can see the blue ones are closer to the mangrove covered islands, so that's the white areas, with the seagrasses to the north, and then the macroalgae and phytoplankton close to the river mouth. So this is suggesting that the mangroves and seagrass material are sources of organic matter to the coastal ocean that are being respired, that are causing this high CO2, that are depleting the delta-13C signature. And further evidence from this mangrove source comes from when we just have a look at our a few other parameters. So we've got our dissolved organic carbon on the top left. We see an increase in that as we increase in high salinity, showing that the river is not the only source of organic carbon to the area. We also see POC increasing, silicate increasing, phosphate and nitrite. So nitrite, phosphate, DOC, POC increases would be suggestive of remineralization happening. And silicate increases at high salinity, so would be indicative of another source of terrestrial material. So normally we use silicate as a tracer of terrestrial material from rivers, but in recent work has shown that mangroves can also be a source of silicate. So this is providing further evidence that the mangroves are a source of silicate and therefore organic matter and inorganic carbon to the coastal ocean. So now just to summarize, apologies. So this is the same schematic that I had earlier, but now I have filled it in with different processes that are occurring. So this is suggesting, the data so far is suggesting that the marine ecosystems here are actually driving the fluxes of CO2 and pH, and it is not the input of river. The river is bringing in this organic matter, inorganic carbon, but the majority of the CO2 is outgassing as it enters the marine environment. We will have some remineralization happening, which releases phosphate, DOC, nitrate into the marine environment. But a lot of what we're seeing is that there is DOC and POC, so organic carbon, along with inorganic carbon coming from the mangroves, which is then being respired and outgassed. But the seagrasses are mitigating this CO2 outgassing. So without the seagrass beds and without the photosynthesis that is occurring, we would likely see higher fluxes of CO2 in this environment. And thank you. That is me. If you have any questions, I'll answer a couple now. If not, we can discuss more at the end.
All right. Are there any questions currently for Sarah? Remember, you can put questions in the Q&A box, and you also can type them to, or you can raise your hand and unmute yourself as well. All right, but if we don't see any questions now, we can take them at the end as well. Oh, here's one. For Sarah, what type of measurements did you make for this work? Sensors and discrete?
Great, thanks, Natalie. So the majority of this work that I've shown today is from discrete water samples. We did collect it alongside sensor data using an autonomous surface vehicle, which I showed with the salinity plots at the beginning. And then it was discrete water samples for organic carbon, inorganic carbon, total alkalinity, delta 13C, nutrients, so a whole host, and oxygen. So the majority of these measurements are discrete samples.
Awesome. Thank you for sharing, Sarah, and thank you for your question, Natalie. All right, so next I'm going to introduce the next speaker, Margaret Ogundari, who is she is a lecturer at the Department of Marine Science and Technology in Federal University of Technology in Accra, Nigeria. And Margaret's project is sponsored by the Ocean Foundation through the Global Ocean Acidification Observing Network peer-to-peer program. The project is aimed at monitoring the coastal ocean acidification during dry and raining seasons in the Gulf of Guinea on the Ilahe coastal region of Ondo State, Nigeria, to provide baseline data. It involves discrete sampling for total alkalinity, the measurement of the surface ocean partial pressure of carbon dioxide, and to calculate the pH and dissolved inorganic carbon from the pair. Margaret, you are now able to share your screen and take it away for your presentation.
Yeah. Sorry, I'm just trying
Margaret,
to
do you
get some
see?
things right. Yeah.
Okay, we can hear you at least. Awesome. Let me see.
All right.
So we see
Thank
your
you,
your
Kalina.
presentation. Yeah.
Again, yeah, okay.
Perfect.
Okay. Thank you so much, Kalina, for the introduction. So, again, I'm Margaret, a lecturer from the Department of Marine Science and Technology in the University of Federal University of Technology, Akure, Nigeria. So as Kalina has rightly said, this project is sponsored by the GOA-ON, that is Global Ocean Acidification networking under the peer-to-peer program. So, and is to provide like capacity building, then also provide baseline data for the OA database, because this side of the Atlantic Ocean, which is the Gulf of Guinea, is very well understudied in terms of the ocean acidification data. So I'm going to give what I'm going to give are just like preliminary results. We are still working on them to be able to explain the nature and the drivers of what we are seeing or observing. So just go along with me as I present a baseline coastal ocean acidification data from the Gulf of Guinea. So I have the picture or the image of the Gulf of Guinea here. It is situated in the eastern equatorial region of the Atlantic Ocean, as we can see. And yeah, I don't know what's happening to thing. I'm sorry, trying to see. Okay. So, and the region is a location for equatorial dynamics and a whole lot of carbon dioxide budgets in the tropical Atlantic, because of the equatorial like there's upwelling. We have major upwelling from the equatorial region, and Gulf of Guinea is part of them. And because of these dynamics, it's a major area for carbon dioxide budget within the tropical Atlantic. So there is occurrence of seasonal upwelling, both coastal and equatorial upwelling, which actually impacts on the biological resources and is the base of a lot of biodiversity that are present in the region of Gulf of Guinea, so that we have a lot of fishing activity and some other activity that are very basic to economical growth of the country. So as I've said before, because of the dynamics of the upwelling, we have it as a region of carbon dioxide outgassing, that is release of carbon dioxide into the atmosphere. And you can see here, these are the different types of the different currents that contribute to these upwelling processes that takes place in this region. We have the different currents coming from the west and coming up at the coast and the equator region of the Gulf of Guinea. Then it is also an area of maritime area of strategic economic importance, just like I said, mostly because of the marine biodiversities, and also especially because of the rich richness in hydrocarbons. So it is a very special area for the maritime activity because of these hydrocarbon activities and also then the biological resources available. So what are the parameters and the sampling that was involved? So the sampling was done during the dry and raining season. The dry season was done in January, and the raining season sampling was done in May. And we have two sampling transects. One on, we call it that is the name of the region, Abereke, one on this axis, and the other on this axis called Awiye axis. And these transects are along a kind of channel because both of these axes open into the sea. So they are a kind of channel with population along the side, and it opens into the sea. So this channel were transacted, and during the dry season, we're able to go into the sea and also took samples along the coastal waters, along the seaside. So one of the parameters is the fugacity of carbon dioxide taken at sea surface temperature. And the method was based on the continuous underway sampling, that is the continuous reading or measurement taken based on equilibration of the atmospheric carbon dioxide with that of the water. Then it is the equipment we use is a locally made equipment that is made for surface water carbon dioxide sensor. Then total alkalinity sampling was done also, but discrete sampling. It was done, and the analysis was done via potentiometric titration. Then for the dissolved inorganic carbon and pH, we calculated it from the pair of the FCO2 and the TA. That's the sea surface FCO2 and the TA, using the carbon seas Excel software that it's normally used for calculation of the carbonate parameters. So definitely the carbonate parameters, the main carbonate parameters are 4 of them: the FCO2, the total alkalinity, dissolved inorganic carbon, and pH. But most times these 4 parameters are not measured to describe the carbonate system most at times. But once 2 of them are measured with the physical properties of the region, we can always calculate the remaining 2. So in this case, we measured the FCO2, that is the carbon dioxide, and the total alkalinity. And from the pair of it, we're able to calculate the DIC and the FCO2. So I have the results. If we look at the results here, we are having the salinity and the SST distribution. So here we can see that generally we have high salinity during the dry season, because like I said, the sampling were done during the dry season and the rainy season, and the set 1 represent the dry season, then the set 2 represents the rainy season. So looking at the distribution generally here, we can see that we have high salinity during the dry season, though mostly it is obvious around the Abereke axis. We have, it's not really obvious along the Awiye axis, but like I said, is this just the preliminary result? We are still looking into the results to be able to understand the drivers of the distribution we are observing. Also, the salinity variation will depend on the station along the channel, because if we look at it, we have this, like I mentioned about the sampling along the channel and then along the coastal waters in the seaside, we'll see that some of the data along the channel, which generally were supposed to be of low salinity, we'll see some of them having higher salinity, even more than the salinity in the coastal water along the open ocean. So all of these are things we are going to look into to find out the drivers of this distribution. But generally, we have higher salinity during the dry season over the rainy season. Also, we have higher temperature, the sea surface temperature during the dry season over the rainy season, and also it's obvious in Abereke axis, but along the away axis, we'll still have to look into the drivers of variation, because it's not like pinpoint that, okay, we have it's higher during the dry season and lower during the rainy season, which is what we expect to happen generally. So here we have the total alkalinity distribution. Also, the dry season set 1 and the rainy season set 2. Also here, we see that we see the ranges of the values of alkalinity. And generally also, we can say that we have a higher salinity during higher total alkalinity, TA, during the rainy season, which is obvious in both the Abereke and the Awoye axis. So, but just like I said, this is also a distribution that maybe we will expect that we should have a lower TA during the rainy season because of maybe intrusion of fresh water or all of that. But we are seeing something that is different, and which actually, though I'm not going to show that one here, but in the property to property correlation, I checked the correlation between the total alkalinity and the salinity in some of the region, especially in the Abereke axis and the Abereke axis set 1 during the dry season, and also in Awoye during the rainy season. It showed a negative correlation, whereas it's supposed to be expected to have a positive correlation. That's between the total alkalinity and the salinity. And of course, the drivers of variation, we are still looking into it, and which is the reason why I did some of this correlation to check, to see, okay, what could be driving the variation we are having here. So also, like I said, generally the values are higher during the rainy season than during the dry season. So for the FCO2, that is the sea surface fugacity of carbon dioxide, I have the plot here showing the distribution of the SCO2. And then I have the plot, the lower panel, showing the difference, that's the gas exchange value, which is given by the difference between the sea surface FCO2 and that of the atmospheric FCO2, giving us like the gas exchange value. And what we have here, these are the values, the range of the values we have at the different axes. And if we look at the range of the values compared to the atmospheric carbon dioxide, we'll see that what we are expecting for the DFCO2, all of it, the results for the DFCO2 are positive. And positive DFCO2 means source of carbon dioxide from the ocean into the atmosphere. And that's just very few, just like two stations, I think, that we are having a negative value for the DFCO2, which is why we are having the 0 value here. And that we have some values below the 0, but they are just 2 points. But generally, generally here we are having all of the station giving us a station for sources of carbon dioxide into the atmosphere, which actually corroborates with what has been identified as the region because of the ocean dynamics, which is the upwelling processes that brings up more of dissolved carbon dioxide to the surface. So I think this is corroborating that at least that we are having a source of carbon dioxide almost all through the transit of study. So now we look into the calculated dissolved inorganic carbon and the pH. But for this now, we're able to just get this calculated result for the dry season. And this is because the sensor, which we use for the measuring of the FCO2, that is the underway sampling, became faulty. It was not taking a good measurement again, so we were not able to use it during the sampling within the rainy season. So the calculation for the dissolved inorganic carbon and pH only took place for the dry season because we were able to get both the FCO2 readings and the TA sampling readings. So, and we have also the ranges displayed here. We are also seeing that we are having some high values of the DIC here, corresponding to some low values of pH. But the expected correlation here, or the expected result here, we should be having, where we have high values of DIC, we should be having low values of pH. But here, if we look at the away axis, we are having B11 is a very high value of pH, which is they are almost of the same value with B13. But we are having B13 to correspond to a lower pH, which is expected. But look at B11 is corresponding to a higher pH value. So all of this distribution, it has to be looked into because this variation depends on the station, and if I be able to say, like Sarah just presented about the mangrove, and she was talking about the mangrove as source of DIC and also source of pH in the environment. So it may be that there are other drivers introducing these parameters other than the known parameters or the known relationship that is expected to take place, or the known sources of these parameters. So, but generally we have lower DIC and higher pH along the channel. That is the general distribution we are having along the channel. We have lower DIC and higher pH, which is the expected distribution. But looking at the coastal waters along the seaside, we are having the opposite because we are having high values of the DIC, and yet we are having a higher values of pH instead of lower values of pH. So in conclusions, what we have been able to, or we can say from these preliminary results we have shown, is that the region is actually a source of carbon dioxide to the atmosphere, and then the seasonal variation in salinity and temperature, as we have observed. Then we have the spatial variation of total alkalinity and dissolved carbon dioxide may be influenced by the mangrove around, which there are some mangroves like this spread along the channel, which potentially we can say they may be influencing the variation, so that we are not as the normal factors, or maybe like the salinity we are talking about. So this is my presentation, and again I want to appreciate the Global Ocean Acidification Observing Network under the peer-to-peer program for provide this baseline ocean acidification data for the Gulf of Guinea. And I also want to thank the management of FUTA and my colleagues in the Department of Marine Science and Technology, and the list of the students who were very instrumental to the sampling, especially during the dry season. They benefited the capacity building aspect of the project, and were also very instrumental to the sampling. So that's my presentation. Thank you all for listening.
Thank you, Margaret. That was amazing. We are running a little bit tight on time right now. So for the sake of that, I think we're just going to keep moving with the presentations and jump right into ISAs. And then any questions for Margaret we can have during our wider group discussion at the end. So next up, we have Isa Elebede. Dr. Isa Elebede is a foremost authority in understanding the role of carbon in ocean research sustainability, significantly impacting our comprehension of carbon dynamics within the oceans and their critical role in climate regulation. He also co-leads the GOA-ON Blue Planet Fisheries Working Group, co-chairs the African Network of Deepwater Researchers, and plays a pivotal role in the Deep Ocean Stewardship Initiative. In the field of ocean carbon research, Dr. Isa's pioneering work reshapes our understanding of this crucial component of marine sustainability, contributing significantly to a more climate resilient and sustainable future. Take it away, Isa.
Yes, sorry, can you see me clearly?
Perfectly.
Okay, great. So can you see my slide now?
Yes, we can see your slides.
Okay, great. Thanks so much for the introduction. I am Dr. Isa. I'll be having a discussion that really concerns a lot of us. Some of us might not be in the global south. However, the global south and the global north have a lot in common, and we need to protect this ecosystem together. So today I'll be talking about the ocean blue carbon and their role in sustainable development, especially in the global south. Okay, yes. So this is my outline. So I'll be talking about the background and as well the value of the ocean blue carbon, and as well the relevance to biodiversity and ecosystem services, including the context of livelihood and economic benefit, and as well the resilience to climate change. And we'll dwell deeply into briefly into some projects in the global south, and as well talk about the challenges people are facing in the global south in terms of managing the blue carbon resources, and how do we balance them, including exploring tools like education and awareness. Then we conclude. So we all know that climate change poses one of the most challenging, and that is why this concept of the oceanic blue carbon has emerged as a way to be able to ensure that there is sustainable development in the global south. So this area looks around the aspect of carbon sequestration, as especially around the coastal and marine ecosystem, and to see how they are addressed and how climate change can foster sustainable development, especially in the vulnerable region. So now, Global South are facing challenges. They are facing multiples of issues, ranging from poverty, food security, to climate threats, related threats. So that is why it has become important that we have an holistic solution towards this problem. And don't forget that ocean blue carbon provides opportunities to address this complex problem in a more sustainable way. So what are the values of the oceanic blue carbon? Number one, we have so many values. However, looking at the concept of carbon sequestration in the coastal ecosystem, the mangroves are vegetated plants that thrive along the coastal area, and they are good source of sequestering carbon. So their root system, which allows to trap organic matters and allows to have carbon sink in the sediment, could prevent the release of carbon dioxide in the atmosphere. Besides, we have the sea grasses, which are another coastal vegetative resources with exceptional carbon sequestration potential. So these are also submerged, and they trap and store carbon in their biomass and their associated sediment. We also have the salt marshes that locates in the transitional zone in the coastal area, and also serve the purpose of storing and as well trapping organic matter around this area. So now, this significance as well relates to the high carbon storage rate that is compared to terrestrial ecosystem. One of the important aspects of this carbon is that they can store and make use of this carbon at a rate that is better or higher than the terrestrial system. And based on this disparity in the carbon storage between this ecosystem, it is possible that this environment can have as a good deposit of organic resources. So now, another area is the aspect of the climate change mitigation. We all know, for example, in recent time, in the next 1 month, the whole world will be talking about, climate topic, especially in Dubai, in UAE, talking about COP28. So as this CO2 level rise, so the ability to capture this carbon is important. And we know that if this is not done very well, it can have effect on the greenhouse gas concentration out there. So to mitigate this climate change is a responsibility from everyone if we want to truly achieve the sustainable development that we so desire. And knowing that in terms of climate change mitigation, the ocean blue carbon can contribute to coastal resilience to be able to stabilize global environmental parameters, such as the temperature, the issue of surrounding sea level rise, and also mitigating the severe extreme weather events that occur. So now, we want to look at the biodiversity and the ecosystem services that exist around these resources. Now, these resources serve as nursery grounds, especially the mangroves and the sea grasses, and they provide shelter for aquatic organisms like the fish, which assist in their growth and survival. Another aspect is the aspect of being able to act as an ecosystem engineer to be able to shape and stabilize the ecosystem. and also to serve as a repository for all biodiversity, so which are home to various localized and unique species. So besides, fisheries as well is another great area where these resources play a good role. Like we all know that coastal ecosystems serve as a primary fishing grounds for fisheries, whereby, if well managed, can add to food security, and as well they can also reduce the effect of overfishing. Then aspect of storm protection, and this system can as well serve as barriers to reduce impact on tsunamis and storms like that, and also filtering water resources in terms of the vegetation, and as well to have the capacity to be able to filter water so that clean water can be accessible to coastal communities. So the implication of this to the global south is that, number one, is aspect of livelihood. Humans that dwell around coastal resources will be able to support their livelihood, and as well, which will benefit them. Then resilience, when their coastal infrastructures are protected due to managing these carbon resources, this will serve as a way whereby the community can always dwell better. And apart from this, there can be many economic resources that can be created from this through ecotourism, aquaculture, sustainable use of all these resources to reduce poverty, because the global south is known as one of the poverty endemic region. So while managing these resources around this region, there will be what enhancement of well-being, and there will be more economic value for the people. So besides the aspect of traditional knowledge, so the generation of these coastal resources, they have a lot of local traditional knowledge which can be passed down to the communities, and this can help in managing these resources, including their cultural values, because this ecosystem also hold cultural and spiritual values for some communities. For example, in some part of Nigeria, some deities use them for spiritual purpose. If you go to Brazil and some other areas, they use all these resources for to better their cultural values. But apart from this, we have the aspect of the communities benefiting from fish resources. Then mangroves serving as an ideal area to implement cultures like shrimp farming, cage cultures, and the rest. And whereby recently I was in Malaysia, where last 2 weeks I was in Malaysia, where we've seen how a guy from the coastal community has been able to implement a shellfish culture, cage culture, and which has attracted a lot of tourists from all over the world. So the act of reducing poverty in the global south is very, very critical. These resources can help to reduce poverty by creating income from aquaculture and creating income to better their education and healthcare facilities. And as well, it has the capacity to create more food, more seafood resources. And from these resources, there can be food security in the region, and families can have access to high-valued nutrition. And the aspect of what? Resilience, which is very, very important to protect their lives and properties. So in the aspect of resilience, we have the natural buffer against sea level rise, and sea level rise including storm protection, and as well issues surrounding enhancing community resilience in the global south. So with this, with resilience to climate change, we can reduce erosion, disaster, and as well making resources available to everyone. So let's quickly look at some projects in the global south. In Mozambique, mangrove restoration activity was done, and as well in Asia, a seagrass restoration was done, which initiated the increased carbon sequestration. This project was able to allow locals to support allow locals to maintain local fisheries and support their well-being in the society. So we have the aspect in Europe and as well in Belize in America and also in South Africa in Ecuador, which has promoted sustainable shrimp farming. So the challenges we are facing, which is very critical to this discussion, number one is that, for example, in Nigeria that I am from, you realize that coastal development are increasing, which the government are not looking at how they can protect these resources. There are issues around land reclamation, urban expansion, which we need to work on. Pollution from all this, our industrial activity carries chemicals to the ecosystem, and as well, the rising temperatures also are big issues. So with this, we need sustainable practices, we need good frameworks, and as well, good community involvement. And there is need for education and awareness, like this platform, this forum we're working on. So we need community education, global awareness, partnership between the global south and global north in the context of SDG 17, more scientific research and more policy advocacy. So in conclusion, it is important that the ocean carbon is critical to sustainable development. And by mitigating climate change, supporting economic activities provide livelihood for the well-being of the people living around the ecosystem. So to protect and to be responsible to manage these resources is very critical to everyone, and there is need for partnership between the global north and the global south to have an effective sustainable management of the blue carbon resources. Thank you so much. Sorry for taking your time. Thank you.
Thank you, Issa. That was a wonderful presentation. But as I mentioned during the last presentation, we're going to save all questions to the end to ensure all of our speakers have time to give their full presentations, as we are running a little bit behind. Our final presentation today will come from Emily Hammermeister. Emily is a third-year PhD student at the National Oceanography Center in Southampton. Emily, please share your slides.
Sorry, I was having some technical difficulties. I'm going to reshare it now. Is that all right?
Of course.
Okay. Let me know if you can see it.
We can see it perfectly.
Okay, all good. Thank you. Sorry about that. And thank you for the nice introduction. Like Austin said, I am a third-year PhD student at the University of Southampton. I'm based at the National Oceanography Center. And today I'm going to discuss with you some of my work regarding novel technologies and applications for ocean acidification observation. So just so you know what to expect, I'm going to give a high-level overview of the marine carbonate system and its relationship with ocean acidification. And then I'll give a little background on the types of carbonate sensors I work with. And I'll briefly discuss some of my ongoing projects that I've used more moving vehicles as well as moored stations for ocean carbon observation. So to start off, the marine carbonate system is considered one of the most important components in chemical oceanography because of its role in the global carbon cycle and regulating the Earth's temperature. And as most of us know, the ocean acts as a major natural carbon sink because it absorbs over a quarter of the atmosphere CO2. As that CO2 dissolves into the ocean, it reacts with the seawater to form carbonic acid that then further disassociates into bicarbonate and carbonate ions. This also releases hydrogen ions, which can protonate the seawater and can affect the seawater's acidity or pH. pH is a parameter we can measure, but it is dependent on temperature, pressure, and salinity. However, there are other components in this marine carbonate system, like DIC, which is the sum of all concentrations of dissolved inorganic carbon, and total alkalinity, or TA, which is related to the charge balance of seawater, and it is commonly described as a measure of the seawater's ability to buffer pH change. Both TA and DIC are measurable and are total quantities, so that they are independent of temperature and pressure. The final measurable parameter in the marine carbonate system is the partial pressure of CO2, or PCO2. PCO2 is the measure of the concentration of carbon dioxide gas dissolved in seawater, and can tell us a lot about CO2 fluxes at the air-sea interface and how much CO2 may be going into the ocean to react with and potentially acidify the seawater. So it's important to note, this is what Margaret was saying as well, between these 4 parameters listed, the carbonate system can be fully constrained so that any 2 of these parameters can be used to calculate the remaining 2. Of course, depending on the process you're looking at, some combinations may be more favorable than others. So with any 2 of those variables, you can calculate non-measurable parameters like the calcium carbonate saturation state, denoted as omega. So the saturation state has been deemed a requirement to adequately monitor changes in ocean chemistry because it's a measure of the capability of seawater to dissolve calcium carbonate. For example, if omega is greater than 1, then the seawater is supersaturated, which promotes inorganic precipitation. And when omega is less than 1, then the seawater is undersaturated, which promotes inorganic dissolution. We care about calcium carbonate because it is a major sink for dissolved carbon in the long-term global carbon balance. And also calcium carbonate minerals are building blocks for the skeletons and shells for countless marine organisms like coccolithophores who photosynthesize, shellfish, corals, and so many others that are threatened by ocean acidification. So I'll skip the gory details, but as we are all very well aware, we have a CO2 problem. Industrialization has caused an extreme increase in atmospheric CO2, which, as I just went over, ends up in our ocean. And as much as our ocean has and will continue to try to buffer the effects of CO2 on our climate, like global warming, it too is being compromised as a result, and our ocean is acidifying. In return, with a changing pH, the marine carbonate species equilibria shifts. So you can see on this plot that even the slightest decrease in pH there can disrupt the carbonate system and result in major decrease of carbonate availability in the ocean. So by better understanding the dynamics of the marine carbonate system, we can better interpret and predict how ocean chemistry and marine ecosystems will continue to respond to a changing atmosphere, ocean acidification, and other factors related to climate change. However, the scales needed to constrain all parameters of the ocean carbonate system, processes, and subsequent ecosystem effects continue to pose a significant challenge. Higher resolution data in larger spatial and temporal ranges, which is highlighted in this figure, are required for better ocean observation to provide accurate and usable carbonate characterization data. Luckily, advances in autonomy have begun to offer newer, more efficient methods for data collection, like some of the available technology highlighted in this table. But as you can see from both the figure and the table, there is room for us to grow to try to fill some of those observational capability gaps. So then this brings us to some of the sensors I work with and develop at the National Oceanography Center. They are what is called lab-on-chip sensors, which is basically what it sounds like. They utilize microfluidic dynamics and are able to perform laboratory wet chemical analysis in situ, including spectrophotometry, single point titrations, and onboard reagents and reference standards. The schematic is an example of how the pH sensor operates. You can find a detailed article in ESNT linked below. And then on the right is just an example of these types of sensors in a deployment setting with their reagents and housings all assembled. So one of my projects was formed to demonstrate a carbonate chemistry autonomous sensor system on board a moving vehicle, a marine autonomous moving vehicle in an operational setting at sea. So we integrated 3 lab-on-chip sensors onto the aft bay of Knox Ferry on Boating McBoatface, which is also known as the AutoSub Long Range, or ALR for short. So TA, pH, and DIC were sensors on board, and they operated alongside a central sensor hub that took data from the CHD on board the ALR and linked it to the sensor's data. Our study site was just off the coast of Southwest England. The black lines are the transects that the ALR followed, and the red triangles are corresponding CTD casts that we did to help get discrete co-samples to help verify our data. But today I'm going to focus on the ALR data that sampled the Celtic margin and continental shelf shown on the right. the transect followed a stair-stepping pattern and a diagonal trajectory, so it never overlapped its own path, but it also wasn't aligned with a single latitude or longitude. So it was kind of a complicated transect. To get a better look, here's an attempted animation to show you this was a relatively complicated sampling method since our data was gathered on a moving three-dimensional plane with sensors measuring at different frequencies. So to analyze this pretty unique data set, I decided to use a standard gridding method where I took a set area between depth and distance traveled that had regular data from my pH and TA sensor. This ended up being about 150 kilometers of lateral distance traveled and up to 80 meters deep, which you can see highlighted green in the figure on the right there. So I ended up gridding that sensor data to see the pH and TA water column patterns along the transect. This is a common oceanographic practice. It's just usually from C2D data and not autonomous sensors on a moving platform. So this is cool to get. We were still able to verify the measurement ranges with C2D discrete sampling as well. But this is cool that this came entirely from autonomous vehicles and we were able to pick up on some of those patterns. It's also important to note that there is a break in the axis on these plots. The shelf is not at 80 meters, it's a lot deeper than that. So like I mentioned before, with any two carbonate parameters, you can characterize the system. So by standardizing the sample region and gridding the data, I could then perform those carbonate calculations using the grids to generate water column data for the saturation state and PCO2 shown in the figures on the left. In turn, this gives us a good idea of what was going on in the water above the shelf's edge during this time. We can see a likely bloom near the surface between 40 and 90 kilometers along the track, and agreement between all parameters, including dissolved oxygen. So basically, this is a really cool new application of autonomous sensors and how to potentially be able to work with our data to get the kind of parameters that you want to look at and what they mean for the surrounding biogeochemical processes. So then, switching gears briefly, I'm also currently working out of Saudi Arabia right now, doing some more deployments on coral reefs in the Central Red Sea. The aim for this project is to show another application for these new autonomous carbonate sensors by looking at reef chemical dynamics, and with the hope to measure and calculate net community production and net community calcification. This is done by looking at the difference in seawater properties from a reference zone off the reef and then in the reef after the water has passed through basically a large volume of calcifiers like corals and Crusoe's coralline algae, or CCA. So you have to know the predominant currents in the region and where the water is coming from. This reef is a great example because for this site because it's an offshore reef and it's pretty offshore, and the water flowing into it is proper open water that has not yet really been exposed to other reef or shore processes. So you can kind of see that from the blue arrows. So I've literally like 2 weeks ago, not even, deployed sensors in both my reference site A on the reef slope and in site B in the reef lagoon. So at site A, at about 15 meters, we have an automated discrete water sampler that will be analyzed for total alkalinity. There's also a SAMI pH there to get a pH gradient across the reef, and then acoustic Doppler current profiler, or ADCP, dissolved oxygen, and a CTD. And then at site B, at about 1.5 meters in the reef flat, is my high frequency sensor package measuring TA and pH using our lab-on-chip sensors. We also have dissolved oxygen, CTD. We're using a SAMI PCO2, and a seabird eco triplet, which measures fluorescence, backscatter, and CDOM. So the idea here is to be able to measure the change in TA and dissolved oxygen from off the reef to on the reef, which will hopefully lead us to community calcification and community production. And a quick note to go check out our very own Sarah Cryer's just new paper. She used this exact frame with pH and DO on it at Caribbean reefs to look at the pH-DO relationship. So a lot of the work I'm doing in the Red Sea is building off of that work as well. So again, here's just our first 5 days of data. I got this back just a couple days ago. So far it's looking so good with our preliminary data. Obviously I will keep you posted, but we're seeing really good signals in pH, dissolved oxygen, and temperature with, during the day, what we would expect to see. And it's going to be really interesting to get our TA data back as well as our off the reef data as well. This is the plots I'm showing you now is just from the high frequency measurements on the reef the first couple of days. So to conclude, ultimately, high-resolution data can enable us to pick up on those smaller scale processes like localized changes in pH and CO2 fluxes in a more cost-effective, efficient, and sustainable manner. It's important that we can use this sort of observing to hopefully uncover more of the oceans or reefs nuances that may be otherwise overlooked. This can significantly improve our understanding of the marine carbon cycle and its interaction with other ocean processes, without as much contribution to the very problem that we are actually seeking to understand and mitigate, which is climate change. So hopefully this work can also help to establish more ways of being able to work with these types of data sets and inner comparisons that can eventually hopefully help lead to build better models and climate models and stuff like that. So with that, thank you everyone for listening, and I'll have questions I think when we're doing the panel. So appreciate it.
Thank you, Emily. That was excellent. Fantastic presentation. As you set up that segue for me excellently, we're going to move into a bit of a panel discussion now. So I want to remind everyone that the chat function down below is an excellent way to put your questions to all of our speakers, as well as to talk about ICONEC generally as a whole. We have about 10 minutes to have these questions. I will read them. But to get us started, I have a question for Isa. Isa, you were talking about blue carbon ecosystems and specifically looking at ways to do some ecosystem restoration. And I was wondering if you could talk a little bit more about how you can balance ecosystem restoration against species climate refugia, where they're migrating away from the equator to stay with their natural habitat conditions. So what's the balance there between restoring past, or what an ecosystem looked like in the past, and potentially an adapting species?
Yes, thanks so much, Austin. Yes, I've known that ecosystem that we have, especially the oceanic blue carbon resources, we all know that they serve as a lot of benefit to humans, especially protecting organisms. So with this, for organisms that are moving from the equator, so there's balancing them requires a lot of work and a lot of activity, particularly, for example, in some part of in Nigeria, for example, some of these ecosystems are being degraded and they are affecting the fishes in terms of capturing. So it is important to explore how we can restore these resources, and as well having proper awareness and working tightly with the policymakers, because some of these coastal areas are degraded because of urban expansion and as well pollution particularly. So there is need to work holistically with all stakeholders and see with scientists and doing more research. Another thing I must say, particularly what Dr. Margaret said, in this part of region, there are limited research, scientific research in this part, especially in this the Gulf of Guinea. So we want to as well use this medium to call on potential support from the global north to see how we can collaborate of research and collect more of data. And some of the degraded ecosystem, to see how we can restore them with partnership, yes. And in this context, because my talk center more on SD, the sustainable development, there's need to work on the SDG 17 that talks about partnership between the global north and the global south, so that we can all have the expected result we want from the ocean decade that we want. Thank you.
Thank you, Isa.
Value in the audience has his hand raised.
I think we have Falilu. Falilu has something to say.
Yes.
Yeah, can you hear me, please? Can you hear me?
We can hear you.
Can you hear me now?
Yes, we can hear you, Dr.
Okay,
Falilu.
thank you very much. Thank you very much to the presenters. I would quickly just like to make a very comment on the presentation of Dr. Margaret concerning the kind of, I would say, I don't know what to call it, kind of a counterintuitive interaction between the ocean variables. For example, when we look at the variability between DIC and pH, where you are supposed to have higher level of pH, maybe you are supposed to have a positive correlation between pH and DIC, for example, but in the context of this very local area, for example, you have in a kind of a negative correlation. I think this kind of situations is observed basically maybe when there's some aspect of the study that is supposed to be done. For example, maybe not just the chemistry, for example, maybe biology could be a factor which is resulting into this variation. I must say that this kind of situation is being observed even in some other local studies within the region. So that's highlight the fact that studies have to be made holistically. When we look at chemistry, we're able to look at the biology alongside with the physical variability, for example, mixing, for instance, because the local, the environment in which the study was conducted is between the estuary and the ocean. So, for example, mixing is another factor, tidal mixing and stuff like that. So, the basis of what I'm trying to say is that we would appreciate more support so that when we carry out our studies, we don't only look at the one aspect of it because of logistic constraint. So, thank you very much for your for the presentation.
Thank you, Falilu, for that. Austin, I hope I can go on to say something.
Of course, yeah.
All right. Okay, thank you for giving me the floor. Yeah, so Falilu, thank you for that observation, which is why I put up that image of the mangrove and some kind of the mangrove that we see around, the mangrove swamp we see around the region where the study was taking, where the study took place. So definitely there are other biological factors than the known factor that increases or decreases the pH and the DIC that are actually taking place. And so it's really pertinent, or it's really important that we have the holistic study, as you said. And so as I say thank you again to the peer-to-peer program which provided that opportunity, we still look forward to more support to be able to carry out this study holistically, especially that this is just like a baseline study. So the monitoring needs to continue. It's a monitoring that needs to continue, and also other factors or other aspects of the study, which is the biological aspect, need also to be put in place. So it's a very good observation, and we really need it. I think what is coming up from this session, as I'm saying, even from Dr. Issa, we are all saying that there's need for collaboration. There's need for collaboration. So that collaboration is very, very important in order for us to have holistic study that will provide the information that we actually give the right knowledge about the condition of the environment. So I think I would like to say thank you to Falilu. And again, I mentioned the use of a local carbon dioxide sensor. I'm sorry I didn't mention the person who made it, though his name is on the slide, on the first slide is Dr. Chris Hunt. He has been using the carbon sensor to take readings along the coast where he works. He's a researcher in the University of New Hampshire. So he is the mentor in this peer-to-peer program while I'm the mentee. So thank him again for the opportunity that we are able to use the carbon sensor even in this project. So thank you, Austin, for the floor.
Yeah, thank you, Margaret. That was wonderful. I think we have time for one more question, and I'm going to throw the floor to Sarah, our presenter, who has a question for Emily.
Thanks, Austin. Just a quick question for Emily. Obviously, I know a bit about the sensors, but I'm just wondering what was the frequency of the sampling that you were doing, and then how long were you able to deploy, and kind of what's the optimum length of time you're looking for deployment versus sampling frequency?
Yeah, so the high frequency frame that we have on the reef flat, the inside of the reef, is sampling for TA and pH every hour. And this can last, right now, our only limiting factor on that is our battery, which is lasting about 6 days. But I do have another battery. So what I've been able to do is I just go out there and do a hot swap of the batteries. So essentially it's going to be out there for a few weeks, which is ideal to get longer periods of this high resolution data is kind of the goal for not only demonstrating this application, but also getting better data, right? And then the other sensors I have pretty high frequent, like the CTD is like every 2 minutes, the eco triplet is every 2 minutes. The PCO2 is going to be every hour as well. And then off the shore, the pH is every hour. And the discrete sampling, which is, it's an auto sampler, so there's like bags attached that are pre-poisoned. That's every 6 hours because we're limited by material, basically, of how many bags we can get down there and have the auto sampler rotating through. So yeah, hopefully I'm hoping to have it out there for about 4 to 5 weeks total, if that answers everything.
Right.
Thanks,
Thank you,
Emily.
Emily.
Yeah, no problem.
With the final 2 minutes, I just want to give a quick plug for our upcoming community meeting, which I believe on November 15th. Yes, I believe that's correct. Steering committee members, correct me if I'm wrong, because today we did not have a chance to discuss too much about our ICONEC community as a whole and put all of you on the hot seat and see what you want from us. But that will be a topic that we will be discussing at that meeting. So if you're interested in getting involved, please contact myself, Margaret, or any other member of the steering committee. And if you're just interested to learn more, do the same thing. With that, 17th. Thank you, Sarah. With that, I'm going to pass the floor back to the coordinators for OA Week. Thanks, Kalina.
Yes, and we're just going to close out the session here. So thank you to the organizers and moderator, Austin, and the speakers for such a great session today. And we also would like to thank you as the audience for joining us for OA Week 2023. So please do not forget to consult the website and to register for any other sessions that may interest you. If you would like to stay up to date with the GOA-ON community, consider signing up as a GOA-ON member if you're not already one on the GOA-ON website. And you can also discover more about the Ocean Acidification Research for Sustainability, or OARS, using the QR code on the screen. So thank you all, and we hope to see you at the next sessions throughout OA Week. Thank you.